We now want to turn our attention to how species change over time through the process of evolution. And then also, how does this result in the formation of new species through the process of speciation? And so in this video, we'll talk about IB section 4.1, looking at how species change over time through evolution and the process of speciation. The process of evolution is a theory, but a theory in science is one that is supported by a wide range of evidence. And for evolution, there is lots of evidence and examples of species changing over time. And before we get into looking at how we know this actually takes place, it's important to define the process itself. And so evolution is simply the cumulative change in the allele frequencies of a population's heritable characteristics over time. And so really what is this saying? Well, if we look at a population, can we see the genetic changes, can we see genetic changes taking place over periods of time? And yes, we can. We've seen this happen with lots of different species. An example that you're probably familiar with would be the peppered moths in England during the Industrial Revolution. Elephants and their tusks, we've observed changing the size of their tusks due to poaching. The Galapagos finches are probably one of the most famous species through Darwin's observations. And so this process of evolution of how species changed is driven by the process of natural selection. So natural selection is a process, and it's the driving force, the mechanism that causes species to change over time. And in a later section, we'll look at how does natural selection actually take place. And so you probably heard this before, but Charles Darwin is the scientist who developed this idea of evolution through natural selection. And he wasn't the only person to come up with this idea. Also, the scientist Alfred Wallace, in very similar time frame, also established this idea of species changing over time on his own, independently of Darwin. Darwin published his thoughts and ideas first, and so he receives the credit. And Darwin replaced this idea of another scientist, Lamarck, Jean-Baptiste Lamarck, in the late 1850s, that suggested inheritance was based off of acquired characteristics rather than inherited characteristics. And so Darwin's idea replaced Lamarck's because it was supported by more evidence. So let's examine some of the evidence, ways that we know that evolution takes place. And the first piece of evidence we want to look at is base sequences or genetic base sequences. And this is one way that we can do this is by comparing the same gene in different species. And so if we look at the sequence of A's, T's, G's, and C's, the nucleotides that make up a particular gene, and we compare them within different species, we can really see how they have changed over time. And a virus, whether it's alive, not alive, COVID, the COVID virus is a great example of a thing, not organism, but thing, changing to many different variants. Over the course of those couple of years, we saw COVID change and different variants develop through the process of mutations, and those would sprout up in different parts of the world and then quickly spread if they were advantageous. When we compare genes between species, we can see that the more closely related species have fewer differences in their base sequences. And so the sequences of DNA, if the species are really closely related, then there's going to be less differences because this supports the evidence that as species change over time, they gradually diverge from a common ancestor. And so that they acquire or develop more and more differences in their genetic sequence. And this allows us to be able to compare different species. Another specific example of this process is a particular gene called the Hox gene. And this occurs in many animal genomes, and its purpose is to help determine the body plan or the different sections of the body. The gene itself is found in Cnidaria, a phylum of organisms, as well as all animals with clear head to tail axes. So fruit fly here in our picture would be an example of an organism that has this gene. And the similarities within the gene can be explained by common ancestry. The organisms that have this gene have a common ancestor, but obviously there's lots of different types of species that have this gene, and so through gradual modification, they've developed slightly different functions, which have led to slightly different body plans due to selection. Whatever form of that gene produced results that were most favorable for the environment that the organisms lived in. A second form of evidence comes through selective breeding. And this is one that we as humans are directly involved with, because this is us, humans, purposely breeding wild organisms to produce offspring with the most desirable traits. And dogs are probably the most common and easily recognizable example of this. By selecting particular characteristics that we desire or like within different dogs, and by purposely breeding different individuals together, we can amplify or further establish those particular characteristics. And this is how we now have lots of different dog breeds, and we know that dogs originated from wolves. And so from wolves now we have all of these different breeds. Other examples that we have selectively bred meat and milk from sheep or cows, the use of horses for transport, pets, dogs obviously, cats would be another example. But this is not just animals, it's also crops. There are a number of different crops that we have greatly changed over the course of our agricultural history. Corn is probably the most common example, in that originally corn had very few kernels, which was much smaller in size than what we see in modern day today. The colors were even different of the actual kernels. And so modern livestock and crops, they're very different today than what wild species that they would closely resemble. And this is through the continued selective ideal type and purposely breeding those particular types that we desire, the characteristics that they have. And so this process is called artificial selection, or through the process of selective breeding. And so to summarize, this really has had considerable changes in domesticated animals and crops over relatively short time periods, 12,000-ish years for crops especially. And this supports that there's been major evolutionary changes caused by humans just through artificial selection over the Earth's history. Our third evidence set is focused on structures, specifically homologous and vestigial structures. Homologous structures are those that are similar in structure, position, and development, and they're similar due to a common ancestor, but they may not be similar in function. And a common example of this is the pentadactyl limbs, as we can see in the picture here. These are similar bones, specifically the humerus, radius, ulna, carpals, metacarpals, and phalanges, so all of the arm bones and hand bones. They're similar within species. They have the similar bones, but they're different in external appearance and function. And so what this means is that they indicates that they were inherited from a common ancestor, but the species, through different selection pressures, those structures have changed slightly as they've evolved for different functions. So for example, for a human, we have these arm bones and hand bones, allows us to pick up things, move things. For a cat or a dog, all of their limbs are used for walking. A whale, its fin would be used for swimming. But all of these species have the same bones. They're just used for different purposes. And so homologous structures, although they don't reveal the mechanism of evolution, nor that organisms evolved into present forms, they help to explain how evolution could possibly take place. It would be difficult to explain evolution taking place without the presence of homologous structures and their explanation. And so they show similarity due to a common ancestor, but not similar function. Vestigial structures are those that are present but no longer serve any function. There's no selective disadvantage to have the structure, but there's no advantage to having it. And we actually have, as humans, have a couple examples of these. One would be our appendix. It probably served some function in the past. Not entirely sure what that function was. It still remains because it generally doesn't cause a selective disadvantage. It is possible that the appendix can rupture, and without medical treatment that could eventually lead to your death, but it's not prohibiting humans from being able to reproduce. And so there's no selective disadvantage of it. Whales, as mammals, also have, in some species, have the remnants of a femur or hip bones. They don't use them anymore. There's no disadvantage to having them, but they still remain within some of the species within whales. And so those would be examples of vestigial structures. The opposite of homologous structures are called analogous structures. And these are those that are similar in function, but they differ in the fundamental structure, and they don't come from a common ancestor. And so what this tells us or indicates that the species have experienced similar selection pressures. Something has, within the environment, has selected for the presence of structures that allow similar functions. For example, flying. Birds and insects, not closely related at all, don't have similar structures by any means, but they both have the capability of flight, and that would be due to similar selection pressures. This leads to a process called convergent evolution, and this is the independent evolution of similar features in different species due to similar environments and selection pressures. And a great example of this is seen in flying squirrels in North America, which are mammals, and sugar gliders in Australia that are marsupials. They're marsupials because they carry young in their pouches. They both have the ability to fly, but they are not closely related. They don't share our recent common ancestor. Some other examples, as I mentioned, would be a bird wing or an insect wing or a bat. Humans, we obviously have eyes. Octopus, they also have eyes, but they're very different. They have evolved differently, and we know that's because of the different placement of nerve fibers. They're behind the retina in the octopus, and so they don't, octopi don't have a blind spot, which is pretty cool. Humans do have a blind spot. And so convergent evolution is not two species becoming one, it's just two species that are similar in their abilities. Doesn't indicate that they have a common ancestor, it's just that they've had similar selection pressures. The opposite of convergent evolution we call divergent evolution, where they're diverging from one another, and this is the gradual evolution of different features in two different species who share a common ancestor due to different environments. And so this is would be a result, or homologous structures would be a result of divergent evolution, and this also gives rise to a process called adaptive radiation. As we previously have discussed, a species is a group of populations whose members have the potential to interbreed in nature and produce viable fertile offspring that can also reproduce. They can't do this with members of different groups. They're not able to produce fertile offspring or reproduce at all, for that matter. And so then speciation is the process of forming new species by splitting of an existing species due to some inability to interbreed. That's typically what is causing this to happen, even if they're provided the opportunity. And so this process results in all of the wide, diverse forms of life that we see on the planet, and we're going to talk about how this actually takes place. In order for speciation to occur, reproductive isolation is necessary. And this is the idea that a group becomes isolated from one another in their ability to reproduce. They can't mix up the genes or interbreed with one another in their separate groups. This is reproductive isolation. And so there are a number of different ways that this can take place. Probably the easiest to think of and the most straightforward is a geographical isolation. This would lead to reproductive isolation. And geographic would be some sort of physical barrier that separates a population into two. And so very straightforward example, if we had a river or a stream that formed that separated a population in half, those two groups are separated from one another, they're not able to get across the river and reproduce, and so that would then cause them to be geographically isolated, which then could lead to would lead to reproductive isolation. Other examples may include like a mountain range, ocean, human development, lots of different examples, but some sort of geographical separation can lead to reproductive isolation. Two other possibilities are called behavioral and temporal isolation, and a behavioral isolation would be some sort of behavior. Typically, this is associated with mating rituals, dances, competition, things like that, that would separate members within a group from one another and then lead to reproductive isolation. Temporal isolation is based off of time, and this is often seen with plants in maybe when pollen is released or when birds begin their mating season. Temporal is all based off of time. And so whether it's geographic, temporal, or behavioral, all three of these are forms of isolation that then can lead to reproductive isolation, which is necessary for speciation to be able to occur. Divergent selection is when traits within the populations become more and more different due to different selection pressures. And so they're diverging from one another. And examples of different selection pressures would be like in climates, if there's different temperature or rainfall amounts, predation with different types of predators, competition, there's different amounts of resources And another common example is the lava lizards of the Galapagos Islands, in which the lizards, in one form or another, got moved to different islands and have adapted to those particular islands, and over time diverged because of the selection pressures and become different species. Speciation can occur in two different forms, allopatric or sympatric. And allopatric speciation is the formation of new species while being geographically isolated from the parent population or from one another. And so this is due to geographic isolation. This isolation prohibits gene flow because the population is divided into two. They can't intermix, they can't breed with one another in these separate populations. And so each population experiences mutations, natural selection, genetic drift. All of these factors work independently in these two different populations, and because they're reproductively isolated, eventually leads to, or can lead to, two separate species. And so this is allopatric being the different locations. They're geographically isolated from one another. A good example of this is the Bahama mosquito fish, which is isolated from one another in different ponds, and in ponds that have more predators, the fish have more powerful tails so that they can have a burst of speed to be able to escape the predators. The other form of speciation that can occur is called sympatric, and this is when new species arise within the same geographic location. It's not separated by some geographical feature, and so behavioral or temporal isolation could lead to sympatric speciation because it's all within one area. And this is oftentimes, at least in plants, can be caused by polyploidy, or temporal isolation, or some sexual selection, picking different partners for mating purposes. And a great example of this is seen with cichlid fish in Lake Victoria. Lots of different species are in this same lake that have arose over time through sympatric speciation. And so through all of this speciation process, adaptive radiation can develop. And an adaptation in a species is a characteristic of an individual that helps it to be suited and survive in its particular environment. And so as species extend into new areas, as they spread out, it can found new populations. And if those new populations are not able to breed and reproduce, it can lead to new species, as we've talked about. And so these populations are diverging from original ancestor species. And if this process is repeated, we can see from one original population lots of new populations and eventually species spreading out from that ancestral species. And so this is adaptive radiation. It's a pattern of diversification in which species evolve from a common ancestor and occupy a range of ecological roles or niches. Their jobs in the environment, what do they do, how are they able to do these things to survive? They all begin to occupy and have different ways that they're able to survive in the environment. This produces a lot of biodiversity. This leads to lots of different species. It minimizes the competition between species, and it also allows sometimes closely related species to live within the same area sympatrically. So what happens if two different species are actually able to reproduce? What happens in that situation? And it typically, well, not typically, but sometimes can produce a hybrid. And this would be the crossbreeding of members of different species. Typically, it's done deliberately by humans. And some examples would be a mule, which is a cross between a horse and a donkey. They have different number of chromosomes, but they're similar enough that they're actually able to produce an offspring. That mule has some very useful traits, but it's sterile. It's not able to reproduce, and so it's called a hybrid. A liger is also a common example of this. A Fairchild mule is a type of plant, and it's a cross between a carnation with a Sweet Williams plant, but it's also sterile. It's not able to reproduce. And so if it is possible to produce a hybrid, which it's typically not, they're pretty much always sterile and not able to reproduce. It can happen sometimes naturally if species overlap in their environments, but again, they're also generally sterile. And so it's not, from a biological standpoint, it's not worth the energy of individuals to be able to reproduce and produce an offspring that then can't pass on the genes. It's not worth that energy and that time for them to be able to do that. And so species are separated from one another through the different mechanisms that we've discussed so far. Behaviorally, another example, the birds of paradise in Papua New Guinea have been featured in Planet Earth episodes, and they have all of these elaborate different courtship things that they do, dances, cleaning of their little areas. All of these things help to prevent hybridization so that they are reproducing with only their members of their own species and we don't get hybrids. It's possible that hybrids can be reversed if the barriers to that process have not yet developed, but that usually has to be assisted by humans. Sometimes it's possible for hybrids to be fertile, but that's also very, very rare. Our last section to discuss is focused on abrupt speciation, and this is when the process happens very quickly, rather than taking long periods of time, and this often results in a polyploidy organism, and that's an organism that has more than 2 sets of homologous chromosomes. This is usually as a result of a mutation. There's chromosome duplication, but the cells don't divide, and so they get extra copies of the chromosomes. It's usually found in plants and some asexually reproducing animals. And those additional sets of chromosomes can come from errors in the meiosis process that would create diploid gamete cells. And so it can lead to a couple of different types of organisms. The first we would call an autotetraploid. And this is when the whole genome gets duplicated. So there's 4 sets of homologous chromosomes, so it's a tetraploid. It's got 4 copies. They usually have really low fertility, and there's a mispairing of chromosomes. Genetic changes can overcome this problem, but it's not super common. An example of this would be a sand rock crest that's found in Eastern Europe, as we see in the picture here. Another possibility is a triploid, or tri3, and this is the fusing of a haploid gamete with a diploid gamete, one that hasn't been unreduced. It can grow, but it can't undergo meiosis, and so the organism would be sterile. In some situations, when there's a crossbreeding of 2 different species, 2 sets of chromosomes, 1 from each parent, would combine. Typically, the offspring is sterile unless the parents are really, really closely related. It's possible for it to overcome being sterile if the allotetraploid cells are produced by mitosis and then divide by meiosis to produce gametes. Those allotetraploids can interbreed with one another, but they're not able to reproduce with the diploid parent species. And so this would create a brand new species with a mix of genes from both parents. Lots of different possibilities. Again, we typically see these in plants, and it's a result of mutations that create these different combinations. Plants are able to withstand these different chromosome differences more so than animals. And so now we've discussed how species can evolve and change over time, as well as how new species can develop through the process of speciation.